Assalamu alaikum, my dear students for class 11th. We are dealing with human physiology in the chapter excretory products and elimination. Human physiology is the portion of the zoology paper, and the chapter here to be dealt with excretory products and elimination. Yesterday we have studied about the human kidneys. At least we have discussed the internal and the external structure of human kidneys, and almostly we have discussed the structure of its basic unit, that is nephron or uriniferous tubule, the parts of the uriniferous tubule, Bowman's capsule, glomerulus, PCT, DCT, collecting duct. And today we will go through the physiology of excretion in the kidneys. Physiology of excretion in the human kidneys, or the kidneys of higher animals, the kidneys of vertebrates, especially the metanephros kidney or the metanephric kidney. This is the physiology of excretion. So far the man is concerned, man is metanephric in nature. The kidney is highly developed. The excretory product is the urea. That urea is formed from the degradation or metabolism of amino acids, which are formed from the breakdown or the digestion of proteins, which are the polypeptide chains in nature, made up of 22 different amino acids. So the source of urea are the amino acids. Amino acids are derived from proteins. Urea is formed mainly in the liver and in small amounts in the brain and kidneys. It is released to the bloodstream and is removed by the kidneys in urine. The excretory process may thus be divided into two main events: urea formation and urine formation. Urea formation and urine formation. So far the urea formation is concerned, urea is a chemical product obtained from the degradation of amino acids, and urine is the formation of a solution of urea in water. So far it is urea formation is concerned. So far the urea formation is concerned. Urea is formed in the liver by a cyclic process. That cyclic process is called the urea cycle. Urea cycle, or it is also called as ornithine cycle or ornithine cytosine cycle, because ornithine and cytosine are intermediate amino acids formed by the degradation of proteins. It is also known as Krebs-Henneslied cycle. Krebs-Henneslied cycle. The amino acids not needed in the body are deaminated by an enzyme. That enzyme is called as oxidase. This deamination takes place when the ammonia groups are removed from the amino acids, but this deamination takes place in the liver. There are a large number of methods how do the deamination takes place. The amino acids not needed in the body are deaminated by an enzyme. That enzyme is called as oxidase. Producing ammonia, it produces a toxic gaseous product. That product is called ammonia. This is very toxic and is quickly changed to urea. This ammonia is changed to urea because if the ammonia is allowed to retain in the body, that will be very dangerous for the body. So it is directly converted into urea. 3 amino acids participate in the process. These 3 amino acids include which are participants in this cycle. The first amino acid is the ornithine. It combines with ammonia and carbon dioxide to form cytosine. It combines with ammonia and carbon dioxide to form another amino acid. That amino acid is called as cytosine. And meanwhile, the water is formed in this reaction. Conversion of ornithine to cytosine by the addition of ammonia and carbon dioxide to form cytosine and water. That is why this urea formation is also called as ornithine-cytosine cycle. The second type of amino acid that is utilized, that is cytosine. Cytosine. This cytosine combines with more ammonia. It combines with more ammonia to form arginine. Arginine is another amino acid present in a polypeptide chain. Meanwhile, the water is also formed. In the third stage, or the third reaction involves in which arginine then decomposes to form urea and ornithine. Arginine then is decomposed to form urea and ornithine. Urea and ornithine are formed when the arginine decomposes into urea and ornithine. The enzyme responsible for this reaction, that enzyme is called as arginase, which catalyzes the formation of ornithine from arginine. And the water is also formed in the reaction. Ornithine is set free for reuse in the urea cycle. This ornithine is again utilized in the urea cycle. Why this is called a cycle? This reaction takes place repeatedly. Whenever this reaction takes place repeatedly, that's why it is called a cycle, and ornithine is again and again utilized. This is how the formation of urea or ornithine cycle in the liver cells in human beings takes place. That's all for this lecture.